Everyone, so today I'll be discussing the answers of the 2019 January Edexcel IEA level biology unit 1 paper. So let's get in. Question number 1. Carbohydrates are one of the main types of nutrients. They include polysaccharides, disaccharides and monosaccharides. Part A, Roman numeral 1. Which formula is correct for a monosaccharide? So monosaccharide means one sugar molecule or one single glucose molecule. Then disaccharide means when two monosaccharides are combined together. A polysaccharide means a large number of monosaccharides combined together to form a long chain, and specifically there are 11 or more monosaccharides in a polysaccharide. So now they're asking which formula is correct for a monosaccharide. So monosaccharides, the ratio is carbon to hydrogen to oxygen ratio is 1 to 2 to 1. Therefore, the answer is B because there is n number of carbons and then in hydrogen n 2 times n number of carbons, and oxygen is same as carbon. So B is the answer. Then Roman numeral 2. Name the reaction that joins two monosaccharides together to form a disaccharide. So, two monosaccharides join together by removing a water molecule. So, the reaction is condensation. Every time when water is removed, it's always condensation. Okay. Then, part B. The diagram shows some information about carbohydrates. Which carbohydrate is being described in each of Roman numeral 1 to Roman numeral 5. So they have divided carbohydrates to two, disaccharides and polysaccharides. And then disaccharides are divided into two. So then Roman numeral 1, present in cow's milk. So they are asking what disaccharide is present in cow's milk. So lactose is the disaccharide that is present in cow's milk. Then Roman numeral 2 contains only glucose subunit. So simply they are asking among these four, what is made up of glucose? Only glucose. So it's maltose. C is the answer. And then polysaccharides have been divided into two. Roman numeral 3, energy store in animals. So energy store in animals is glycogen. So C is the answer. Then Roman numeral 4, 1,6 glycosidic bonds present. So B. In amylopectin, it's highly branched. Therefore, it contains both 1,6 and 1,4 glycosidic bonds. So you know when two monosaccharides are combined together, water molecule is removed by condensation and they are combined together. So they are combined together by a glycosidic bond, and that bond is 1,4 glycosidic bond. But if it's branched, a polysaccharide is branched. So therefore, polysaccharides also have 1,6 glycosidic bonds. So here only amylopectin is branched. Therefore, it has 1,6 glycosidic bonds as well. And the ones which have only 1,4 glycosidic bonds mostly have a helical structure. Okay. Okay. Next part. Question number 2. The diagram shows a coronary heart disease risk calculator. So here is the risk calculator. So here we must enter our sex, ethnicity, age, total cholesterol level, HDL cholesterol level, systolic BP, patient taking blood pressure medication, patient has diabetes, patient smokes, and then we must calculate, and then we'll get the risk. Risk means the probability of us getting the disease. Part A, which of the following is a medication to control blood pressure? So for blood pressure, in Edexcel, the advanced word is hypertensive. So if you're having blood pressure, then it's said as you're hypertensive. You have hypertensive. So here, the answer is antihypertensive. Okay, so it's B. Then part B, explain why the risk calculator takes into account total cholesterol and HDL cholesterol levels. So before going to the answer, we all know that when cholesterol level increases, the risk of heart diseases also increase because the plaques, the fatty deposits, they build up in our arteries, then our blood pressure increases, therefore cholesterol level increases. Why the blood pressure increases? Because when plaques are deposited in the arteries, when blood flows, it's the pathway is narrower. Therefore, the blood needs to flow due to the high demand of our body tissues. Therefore, the blood pressure increase because if not, the blood cannot pass through. So this causes high blood pressure. So we know about that. So because of that, we can write there is a link between total blood cholesterol level and heart disease. And also they're asking about HDL cholesterol level. So as we know, HDL is high density lipoproteins, and if HDL levels are high, the risk of cholesterol decreases. The risk of heart diseases decrease because HDLs, they take the saturated fats that are binded to our membranes and take it to the liver and destroy them. So because of that, this decreases the amount of saturated fats in our membranes. Therefore, that means it decreases cholesterol. But they are asking, why does the risk calculator tells us to include HDL cholesterol levels as well? So we don't take HDL alone. We always take the LDL to HDL ratio. So the LDL to HDL ratio is the one that determines, it's taken together. So we cannot take just LDL alone or HDL alone. In order to determine the risk, we need the ratio. So HDL is not a risk factor, yet the LDL to HDL ratio determines the risk level. So that's why HDL is also included. Okay, part C. Explain why the 10-year CHD risk would change if this person ticked the box next to the patient smokes on the risk calculator. So here we can see this particular person has not ticked near the patient smokes part. So they are asking what if the person ticked it and what would happen to the risk. So the answer is the risk would change to increase. So that means the risk will be greater than 6.62%. Because smoking increases blood pressure, because nicotine in cigarettes damage the endothelial, the artery lining. So this causes atheroma buildup and therefore the blood pressure increases. So that's the answer. Okay. So therefore, when blood pressure increases, that means it affects, it links with heart diseases. Therefore, the risk of coronary heart diseases will increase. Okay. Part D, Roman numeral 1. State one other factor that the risk calculator should include to improve the accuracy of the value of the 10-year CHD risk. So I wrote level of exercise, and I'll give you some examples as well. So you can write salt intake, fiber intake, alcohol intake, body mass, BMI, obesity, waist to hip ratio, stress levels, and family history. So that means, like, if your parents have heart diseases, then you're more likely to inherit it. Okay. So I wrote level of exercise because level of exercise also um, links to coronary heart diseases. That means if we exercise more, then the fatty deposits in our muscles, um, they will be removed. So because of that, then the risk of getting coronary heart disease will decrease. But if we don't exercise, that means if the amount of us exercising is very low, then people will develop obesity. Okay. So and then they will have more weight, so that will cause heart diseases. So I wrote level of exercise. Then Roman numeral 2, predict the effect that taking this factor into account would have on the value for the 10-year CHD risk. So as I wrote level of exercise, I wrote when exercise level decreases, the risk of CHD increases. So if you wrote something else, for example, like salt intake, you can write if this amount of salt taken by a particular person increases, then the risk of CHD will also increase. Since they have given us only 1 mark, we can stop from that. But if we take more, if we give it a right explanation, then it would be like, if we take more salt, then our blood pressure will increase. So because of that, the risk of CHD also will increase. Okay, so I just gave, like, it's a extra point, but then you don't have to write like that because we are only getting 1 mark here. But if they're asking, if they gave 2 marks, then we can write. Roman numeral 3. A person used this risk calculator. Explain why the value obtained for the 10-year CHD risk may be an underestimate. So the answer is, not all the risk factors that cause CHD are mentioned in the calculator. So not all the risk factors are included. So maybe a person is not exercising really well, and maybe um, he doesn't smoke, he doesn't have diabetes, but he doesn't exercise. So then there will be a problem. Maybe he's obese. So then it is not entered. Therefore, the risk can be underestimated. Then, most often people underestimate, for example, their body mass or how much they smoke, and also HDL levels or total cholesterol levels or blood pressure can be taken as estimates. So, this happens when we take tests. For example, for checking the amount of cholesterol levels in our body, we take a test as lipid profile. So basically we sometimes get decimal values, and sometimes we doesn't take the decimal value, we just ignore it, or sometimes we just take the whole number. So things like that can happen. Even those small things are just underestimates. So then if we put it to the risk calculator, then they can give another value. So things like that can happen. Even the smallest thing can affect it. Okay, so that's that. Question 3. Nucleic acids include DNA and RNA. Part A. Each single strand of a DNA molecule is synthesized from mononucleotides. Draw a diagram to show two mononucleotides joined together in a single strand of DNA. Use the symbols shown for each component in your diagram. So they have given a phosphate group, pentose sugar, base, and a bond, and 3 marks. So we must show two mononucleotides joined together. So here I have drawn one mononucleotide. So here we have the pentose sugar, and then we have the phosphate group attached, and then here we have the base, and the bonds are here. So we must always remember, like, the next if you are going to draw a chain of mononucleotides, the next phosphate is joined like this. It's always the phosphate that is joined to the pentose sugar in a chain. So you must remember that, because if not you will forget and then draw something else. Okay, and then part B. The diagram represents part of a DNA molecule. The pitch is the length of one complete turn in the double helix. There are 10 base pairs in one pitch. Roman numeral 1. Calculate the distance between one base and the next base on one strand of DNA. Give your answer to an appropriate number of significant figures. So here, pitch of a DNA, that means a complete turn in the double helix. So pitch is 3.4 nanometers and the diameter is given 2 nanometers. Okay. So they are asking, there are 10 bases in one turn, and they are telling us to calculate the distance between one base. So that means we must calculate this sorry. We must calculate this distance. This is the distance, the red line. This is the distance we need to calculate. Okay. So length of one complete turn or pitch is 3.4 nanometers. So the number of turns the number of turns is 10. That means the number of bases in one turn is 10. So to find the distance between one base and the next base, we must divide 3.4 by 10. So then the answer is 0.34 nanometers. So that's the answer. For 1 mark, that's the answer. Roman numeral 2. The diagram shows the structure of thymine and 4 other bases, P, Q, R, and S. Bases P and S can form 3 hydrogen bonds each, and bases Q and R can form 2 hydrogen bonds. Explain which of the 4 bases, P, Q, R, or S, is adenine. So adenine, you must keep in mind always, adenine and guanine, okay, they all are purines. That means they have 2 rings. Okay. Then cytosine, uracil, and thymine, they are always one ring. So they have given an example of thymine. Okay. So they always have one ring, and purine have 2. Okay. So the answer is adenine is R, because it forms 2 hydrogen bonds, and also again R is a large purine base because it has 2 rings, unlike the given example thymine. So now just forget about P and Q, because we know they're asking about adenine. So we know adenine is a purine and it has 2 rings, but P and Q they don't have. So they can be either cytosine or uracil. But we have to consider about R and S. Okay. So it's R because you can see in S is guanine because it has H2N, and then it has H, and also you can remember that it has a double bond, oxygen. And you must remember that adenine doesn't have that. So because of that, it's adenine. So you get the pictures in your textbook. So you need to, like, at least find something that you can remember, like a small information about the picture so that you can remember, okay, adenine has that and guanine doesn't have that. So then it's easy, because or else you have to remember everything and it's hard a little bit, but then you can try. Okay, so that's the answer. Then C, compare and contrast the structure of messenger RNA with the structure of transfer RNA. So both are RNAs, so that's the thing we, at a glance, that's the answer we can write as a similarity. And compare and contrast means you must always write similarities and differences. Okay, so similarities, I have written both contains RNA and both are mononucleotides and both have uracil instead of thymine and has the ribose sugar. Okay, and then differences, I have written mRNA is unfolded straight chain and tRNA is folded and takes a clove leaf arrangement. And also you can write mRNA, tRNA is small compared to mRNA. And then one of the main points you must write is mRNA has codons, okay, and then tRNA has anticodons and amino acid binding site. Okay. Then question number 4. The photograph shows a mammalian heart. The blood vessel labeled X is a branch from the aorta. So this is the branch from the aorta. Okay. The aorta is one of the major blood vessels of the heart. Part A. Explain why it is important that blood vessel X branches directly from the aorta. The answer is X is a coronary artery. Okay, if there are arteries in the heart, that means heart muscle, it's obviously and always a coronary artery. And it carries oxygenated blood for aerobic respiration. So, aorta is the closest artery which carries oxygenated blood. So, it's important for X to branch from the aorta so that it will get enough blood. Okay. Part B. The diagram shows the internal structure of a mammalian heart. Roman numeral 1. Explain which stage of the cardiac cycle is shown in this diagram. So it's atrial systole because the atrioventricular valves are open. Okay, so we can see these are the atrioventricular valves. Okay, so they are open. So because of that, open. So because of that, it's atrial systole. So you can also write ventricular diastole, because if it's atrial systole, then it's obviously the other one is ventricular diastole. For both, they have given marks. Then Roman numeral 2, draw arrows on the diagram to show the flow of blood through the left side of the heart and into the aorta. So this is the left side of the heart. So I'll draw again. Left side of the heart. So it comes from here like this and goes and like this. So this is the aorta. Okay. So this is the pathway. Okay. Comes and goes like this. Okay. So this is how you must draw the arrows. Part C. The graph shows the changes in the internal diameter of the aorta during 2 cardiac cycles. Explain the changes in the internal diameter of the aorta. So they have given a graph. So we can see the diameter increases with time and then again decreases, increases and again decreases. Okay. So they are asking to explain the changes. So increase in diameter due to high pressure and large volume of blood leaving the ventricles. Therefore, the elastic fibers expand and then again a decrease as the elastic fibers recoil again as the blood leaves it. So the due to the high pressure and the large volume of blood flowing through the aorta, the elastic fibers they expand and allows the blood to go and they withstand the pressure. We have learned about that separately, that why there are more elastic fibers in aorta, and also we have learned that close to the heart, the arteries have more elastic fibers, and more the artery goes away from the heart, the number of elastic fibers decreases, because close to the heart there's high blood pressure. Okay, so due to the expansion of the elastic fibers, the internal diameter of the aorta increases, and again we can say it has decreased. It's because as once the blood leaves, the elastic fibers recoil again, and then it again increases, again decreases. Okay, Question number 5. Liposomes are spherical structures composed of phospholipids. They can be made by adding phospholipids to water. Liposomes can be used to study membrane permeability. Part A. The diagram shows a liposome. Roman numeral 1. Calculate the volume of this liposome using the formula volume equals 4 over 3 pi r cube. So we know the value 4 over 3, we know pi, where the value of pi we can get from the calculator. We need to find the radius. So they have given us the diameter as 100 nanometers. So we must divide the diameter by 2 to get 50. That's the radius. Then we substitute it to the equation they have given. V equals 4 over 3 into pi into 50 cube. Then this is the value. So you can either keep it in a non-standard form or standard form, because they haven't told us to specifically um, keep it in standard form, but it is better if you write it in standard form as 5.2 into 10 to the power 5, because most in Edexcel marking schemes they look for standard form other than just leaving the answer. Okay. Then Roman numeral 2. Explain the arrangement of phospholipids in liposomes. So the answer is polar phosphate heads face inward, forming hydrophilic interactions with the aqueous environment or water, while the nonpolar tails face outward away from the aqueous environment and form hydrophobic interactions. So you must remember something, always if something is making an interaction with water, it's always hydrophilic. Okay, and also it's polar, that always interacts with water, forms bonds with water, because polar water is also, we know H2O, it's a polar molecule. Okay, it's a dipole, therefore it's a polar molecule. So the polar phosphate heads, they are also polar, therefore they make interactions with water. Polar polar, non-polar non-polar. So if something makes a bond with water, it's always hydrophilic, and non-polar always it's hydrophobic. The interactions we have between R groups, it's always hydrophobic. Okay. Question number B, part B, sorry. The presence of cholesterol in the membrane affects membrane permeability. A student investigated the effect of cholesterol on the permeability of liposomes to glycerol at different temperatures. Liposomes were made by replacing 20% and 50% of the phospholipid with cholesterol. Liposomes without cholesterol were also made. The graph shows the results of this investigation. So permeability means how fast or how easily the glycerol can pass through the membrane of liposomes. Okay, so here they have taken x-axis as permeability of membrane to glycerol, and then y-axis temperature. So here we can see when there's 50% cholesterol, the membrane permeability is very high, and then 20% the membrane permeability is very low, and 20% it is much high, a little bit higher than 50%. But we can see with no cholesterol, the membrane permeability is really high. And also something we must observe here is temperature. Now we know about the y-axis. We know about the permeability, how the permeability increases. That means with no cholesterol, the permeability is really high. But you must consider about temperature as well, because they have given temperature. So it's really important in Edexcel in papers to study graphs, because if they give two axes, we must always look at both the axes and see how one axis affects the other. Okay, so here temperature, we can see that with temperature increasement also the membrane permeability is high. Okay, so that means with no cholesterol, the membrane permeability is high, but also the temperature also affects. Okay, then question, no, Roman numeral 1. How does glycerol pass through the liposome membrane? So it's B. Why it's B? Because we cannot take endocytosis or osmosis. Just forget about that. The problem is between active transport and diffusion. So at first, at a glance, when I was answering this question, I thought it was active transport, because it's glycerol, okay? It's substances, okay? So I thought it was active transport, because mostly in membranes, between membranes, it's active transport. I just thought like that, okay? But then I just then try to study again and just look at the marking scheme to make sure whether my answer is correct or not. And then only I realized active transport, the main key point is we need energy. It doesn't take place passively. For active transport, we always need energy. So because of that, it cannot be active transport. For diffusion, we don't need energy. It just happen naturally. So because of that, the answer is diffusion, and diffusion is always across the concentration gradient. So this is also across the concentration gradient. Okay, so it's diffusion. Roman numeral 2. Describe the effects of cholesterol and temperature on membrane permeability as shown in the graph. So we took a look at the graph earlier. So the answer is increase in temperature increases membrane permeability. Increase in cholesterol decrease permeability, meaning cholesterol has greater effect on membrane permeability at high temperature according to the graph. Okay. Then Roman numeral 3. Explain why cholesterol and temperature affect membrane permeability. So increase in temperature increases the movement of phospholipids by providing more kinetic energy. So you must always remember, if its temperature increase, it's always linked to kinetic energy. You know, when answering questions about enzymes, it's always kinetic energy. Why temperature increases? The enzymes get more kinetic energy, so they can bind more efficiently with its substrate. So that's it's if it's temperature, it's always kinetic energy. So if it okay, so you must remember that. Okay, because some answers they are always joined together. Even though the question changes some answers, the style we are writing it, it's always same. Okay, then too much cholesterol present reduce fluidity of the membrane and reduce movement and act as a barrier. So when there's too much cholesterol, it's hard for the glycerol to pass through, so it acts as a barrier. That means it opposes its movement. Okay, so that's then question number 6. Platelets are involved in the blood clotting process. Part A. The table shows the phospholipid content of the membranes of platelets. So they have given some phospholipids and then the percentage of total membrane phospholipids and also how they are distributed in the phospholipid membrane. When platelets trigger the blood clotting process, more phosphatidylserine molecules move into the outer layer of the membrane. Roman numeral 1. Estimate the ratio of phosphatidylserine in the inner layer to that in the outer layer before the blood clotting process is triggered. So they are asking about this. Okay, they are asking about this. This is the thing they are asking about. So inner to outer. So inner it's 15. Inner it's 15. So here they have given the percentage of total membrane phospholipids. Inner it's 15. Outer I have taken as 3. Okay, I have taken as there is a for the marking scheme also there are about 3 multiple answers, so you don't have to worry about that. It's just you have to take something very close. So inner to outer 15 to 3, the ratio is 5 to 1. That's the answer. Then Roman numeral 2. Describe the effect that the movement of phosphatidylserine into the outer layer will have on the content of phospholipids in the membranes of platelets. So the answer is will not have any effect on the total membrane phospholipids. High phospholipid content in outer layer and low content in inner layer. So outer layer the content is very high and the inner layer the content is very low. So because of that there is no effect. Um, Roman numeral 3. Describe how the movement of phosphatidylserine into the outer layer results in the production of thrombin in the blood clotting process. Okay, so you know about the blood clotting process. What happens when there's internal bleeding or uh, when our tissues are damaged? It triggers platelets to come out. So when they pass through the collagen fibers, the platelets break and they release thrombin and plasma proteins and the clotting proteins, etc. So the answer is when phosphatidylserine move into the outer layer, this will alter the membrane properties. That means it will damage and this will cause platelets to break and release thromboplastin, an enzyme which converts prothrombin into thrombin. Okay. Then, part B. Thrombin inhibitors are drugs that have an effect on the time taken for blood to clot. Explain why thrombin inhibitors affect the time taken for blood to clot. So, inhibitor means something that opposes something. For example, now here it's thrombin inhibitor. That means it inhibits thrombin. It does not allow thrombin to work properly or not work at all. So the answer is thrombin is an enzyme. So effect of an inhibitor change the shape of its active site. So if it's enzyme, then you must write it changes the shape of its active site. Therefore, thrombin cannot bind to fibrinogen. So, less or no fibrinogen will be converted to fibrin, which leads to less or no fibrin to form a mesh and trap blood cells and platelets. Okay. Question number 7. Some insects lay eggs inside leaves. This causes the leaves to produce swellings called galls. The galls supply developing insects with nutrients and protect them from the external environment. The photograph shows galls on a leaf. So here these are the galls they have marked. Ring-shaped small circles. In an investigation, the concentration of protein molecules and amino acids found in healthy leaves, leaves with galls and in the galls themselves were determined. Part A. The insects were removed from each gall before the investigation. Suggests why the insects were removed from the galls. So the answer is it's unethical to kill animals, so the insects were removed because they contain proteins or amino acids, which will give an incorrect value for the content in gall. So that means they are taking only the amount of amino acids and proteins in galls. But if insects are also there, then the the content of proteins and amino acids of insects will also be taken. It will be an underestimate. So because of that, the insect were removed. Part B, Roman numeral 1. The diagram shows the R group of the amino acids were extracted and dissolved in nonpolar organic solvent. Roman numeral 1. The diagram shows the R group of the amino acids alanine, aspartate, and tryptophan. So they have given alanine and then aspartate and tryptophan. They are asking which row of the table describes the solubility of these amino acids in a nonpolar organic solvent. So the answer is B. More soluble alanine because it only has CH3. Okay. It is easily dissolvable, especially in organic solvents. Then second is tryptophan. Okay. Because now the problem is between aspartate and tryptophan, but then you can take because aspartate has a COO bond. Okay. So it's a carboxylic group. So carboxylic groups they are not soluble. Okay. So because of that, it's tryptophan that comes to second and aspartate comes to last. It's the least soluble. Okay, because it has double bonds with oxygen, especially. So you know, especially the double bonds between carbon and oxygen, they are really strong. Okay. Then Roman numeral 2. The solubility of an amino acid can be determined by measuring the maximum mass of the amino acid that dissolves in a known volume of the solvent. The solubility of the amino acid histidine in a solvent is 43.5 grams per dm cube. The mass of the amino acid leucine that dissolves in 250 cubic centimeters of the same solvent is 5.5 grams. Calculate how many times more soluble histidine is than leucine. So they have given already the solubility of histidine. So we must find the solubility of leucine in order to compare them. So mass of leucine they have given 5.5 grams, but they have given the concentration of leucine. Therefore, we need to find the volume. So it's cm cube. Therefore, we divide 250 by 1000 and then we can get 0.25 dm cube. Then to find the solubility, we must divide the mass by volume. So 5.5 divided by 0.25 equals 22 grams per dm cube. So in order to find how many times histidine is soluble than leucine, we must divide 43.5 the solubility of histidine with the solubility of leucine, which is 22, and we get 1.98. So histidine is approximately 1.98 times more soluble than leucine. Okay, so that's that. I hope you understand how I took the answer. So I've written it by process so that you all can understand how to get the final answer. Now part C, it's an asterisk question. So we need a lot of critical thinking as well as asterisk mark questions are the questions in the whole paper that we get full marks. So it's important to answer this. Okay. So the gall forming insects secrete saliva into the plant tissues. So the saliva contains enzymes that change the nutrients in the leaf and cause the galls to form. Okay. So the graph shows the concentrations of protein molecules and amino acids. Molecules in tissues from a healthy leaf tissues of a leaf with galls and in the galls themselves. Okay, so they have given the protein molecules present in healthy leaf, leaf with galls, and amino acid molecules present in healthy leaf, leaf with galls and galls. So we can see at a glance in these two graphs, the amino acids molecules present in galls are really high, and also the protein molecules present in galls are really high. And also they have given, the table shows the abundance of 5 amino acid molecules. Okay, so the question is, explain the results of this investigation. Use the information in both graphs and the table to support your answer. So we need to explain the results of the investigation. So I have written the answer. There is high concentration of protein in the galls. Then there is high concentration of amino acids in galls because it's needed for protein synthesis, because the enzymes in saliva were breaking down proteins for the use of insects. Then uh, that's about the graph. Then alanine, arginine, histidine are very abundant in galls because amino acids are abundant in proteins in galls. None of the amino acids are abundant in the tissues of leaves with galls because they are in galls. Leucine and tryptophan are not in galls. Okay. Then question number 8, last question. The photograph shows two mammals, an elephant and a mouse. Part A. The height of a mouse is 3 centimeters. Calculate how many times taller an elephant is than a mouse. Use the white line drawn on the photograph of the elephant to calculate this value. So, they have given the magnification of the elephant, which is 0.02 times. So, we need to find the actual height. So, I think you all can remember the magnification equation, image over actual. So this is the white line. So you all can take a ruler and measure it. So I got 4.5 centimeters. So then to get the actual height of the elephant, I divided 4.5 by 0.02 and I got 225 centimeters. Then to find out how many times tall an elephant is than a mouse, I divided 225 by 3, which is the height of the mouse, and which is 75. Therefore, the elephant is 75 times taller than the mouse. Then B. The respiratory system of an elephant is different from that of other mammals. The lungs are attached to the chest cavity wall and diaphragm by collagen fibers. Describe how the lungs of an elephant are adapted for gas exchange. So the answer is, first of all, they have given us two informations. The lungs are attached to chest cavity and the diaphragm by collagen fibers. So first of all, we can use them and comment. So we can tell, since the lungs are attached to the chest cavity, volume increase and pressure decrease. Okay, and then as usual, we know in every lung alveoli are present. So according to the theory, we know we can write alveoli provide large surface area for faster diffusion. Alveoli formed from one cell layered epithelial for small diffusion distance. Efficiency of the concentration gradient is maintained by good blood supply. Okay, and also you can write that as collagen fibers are also mentioned. So as an extra point, I'll tell you, since collagen fibers are mentioned, you can write that collagen fibers, they give they strength, they have a lot of strength. So it helps the lungs, it supports the lungs to do efficient gas exchange. Okay, as it has a lot of strength. Then again asterisk question, part C. Graph 1 shows the oxygen dissociation curve of hemoglobin for a mouse and for an elephant. So here we can see partial pressure of oxygen. Graph 2 shows the mass specific metabolic rate for a mouse and for an elephant. Mass specific metabolic rate is measure of how much oxygen is needed for chemical reactions per gram of body tissue. So we can see the mouse, um, it has a very lower body mass in kilograms compared to elephant, and it has a high mass specific metabolic rate. And then oxygen, it has a really big body mass in kilograms compared to the mouse, and it has a very low mass specific metabolic rate. So the question is, explain the difference in the oxygen dissociation curves for hemoglobin for a mouse and for an elephant. Use the information in both graphs to support your answer. So I have written oxygen dissociation curve for mouse is shifted to the right, and therefore it its hemoglobin has lower affinity to oxygen than elephant's hemoglobin. So you must remember, if a oxygen dissociation curve is shifted to the right, that means the hemoglobin has lower affinity to oxygen. If it's shifted to the left, then always the hemoglobin has higher affinity to oxygen. Okay, so that's a point. Mouse has a higher metabolism rate. So here I have mentioned metabolism rate to this. This is a man specific metabolic rate than elephant, and high respiration rate as it has large surface area to volume ratio. Therefore, efficiently metabolic rate, it has a high rate because it is really small compared to the elephant. So it has its tissues almost near to, but the elephant is big. So because of that, the tissues are far away. Therefore, the rate is slow compared to the mouse. Mouse is more active as it has to escape predators, so mouse tissues has lower partial pressure of oxygen so that hemoglobin can readily release oxygen, whereas it's opposite for the elephant. So another key point you have to remember is tissue tissues should have, if the partial pressure of oxygen is low, that means hemoglobin can readily release oxygen. If the partial pressure of oxygen is really high, hemoglobin does not release oxygen and it takes oxygen. For example, if we take humans, overall the topic of these oxygen dissociation curves, in lungs, oxygen the partial pressure of oxygen is really really high. Therefore, hemoglobin has higher affinity to oxygen and it readily takes oxygen, it attracts oxygen. But when it comes to tissues, our tissues does not have a lot of oxygen, and hemoglobin is the thing that in the red blood cells they go and release oxygen and they give oxygen and other nutrients for tissues to respire. So in tissues we have a lot of like it has a very low partial pressure of oxygen, because then only hemoglobin can release it readily. So this is a point you must understand, a key point, so that when you get questions like this, even if it's an animal, you can always link the theoretical points and write. Okay, so you can join the theory to the question they have given, link it together and then write. Okay, so I hope you understand. So this is the end of the paper. So a thing I want to share is that all the answers given in this paper are Edexcel marking scheme guaranteed. So if you write these answer exactly like this, I mean, if you link, you don't have to like by heart my answers and write. I mean, I'm just giving you a guarantee that you will get full marks, because these are full mark questions. So you full mark answers. So you will get full marks if you write them. So you don't have to worry. And also I also have uploaded a short of this whole video, so that I haven't explained anything in it. I just have uploaded the short. So if you're running out of time, if you want to like just um, go through your answers after you have written it, you can check my shorts and go through paper marking. For paper marking, you can go through my shorts, and yeah, I have written the full answers in full sentences so that you can understand. So please, you all can refer to that. And please subscribe, like, comment, and share my videos with your other study mates so that they can also get something good from my videos. So please do comment if I did a mistake, or if my explanations are not good, so that I can improve really well. And maybe there are some pronunciation mistakes as well, as I did some mistakes throughout the paper. I'm really sorry for that. I'll improve myself, and please do comment positive comments so that I can inspire and motivate myself to upload more videos. And also, I'm also a IA level Edexcel student, so I just thought that when I'm doing papers, it's nice if I can upload videos like this, because actually my mother is the one who gave me the idea. So because explaining like this also makes me also helps me to understand on my own lessons, and helps me to remember my answers really well. So thank you guys for watching my video, and let's meet from another video. And also I'll be uploading other subjects as well, and all the years. So I started from 2019, but it's not like I'm gonna upload biology first then physics. I'll be uploading physics, chemistry as well. So I know physics is really hard, it's hard for me as well, so I'm just trying to go through it. So I'll be putting physics videos as well, because physics is one of the most challenging subject in Edexcel IA level compared to biology and chemistry. So let's meet from another video. So please subscribe my channel so that once I upload a new video, you'll get notifications. So the next batch we'll be getting ready for the 2026 exam. So we'll ace it somehow and succeed and achieve our dreams. So let's meet again. Thank you. Bye. Have a nice day.